Welcome to Hacking the Hustle, everyone. This is your host, Benji Sklar. Really excited to have my friend Joey Wippold on the podcast. What's up, Joey?
What's up? Nothing much. Just having a good day here in Texas.
Very nice. So a little background on Joey. Joey is currently getting a PhD at Texas A&M in biomedical engineering, and he's also working for the Department of Defense in their biotech group, specifically their sensors and electronic devices doctorate. And
Yeah.
Joey and I grew up together. He was a really sharp guy, and now I want to have him on the podcast because I feel like he's at the forefront of the battle against the coronavirus and other viruses that are facing us in the future. So I'd love to just start off, Joey, with what's it like for you to be getting a PhD in biomedical engineering and working for the Department of Defense with the coronavirus going on right now?
Well, yeah, it's getting a PhD in biomedical engineering, as one can imagine. It's a lot of hard work, a lot of long hours, a lot of project planning and running experiments. Working with the Department of Defense has facilitated some of that. They have excellent state-of-the-art facilities that are made available for me to use. They have excellent funding for propelling the projects forward.
Mm-hmm.
So it's a really good symbiotic relationship. I think that the relationship started kind of happenstance that they needed someone with my skill set, and I'm filling a void that they currently we're looking to move into, and specifically that void is high throughput screening systems, and that's what my lab here and what specifically myself specialize in. And so what that means is just like we use microfluidics, so tiny fluid channels, and even sometimes droplet microfluidics, where these mini bioreactors, kind of like nanovesicles you can call them, that you put stuff in them, and you're able to screen the contents for whatever biochemical reactions on the fly. And so why is that important for, in medicine or in the Department of Defense? I'll address the medicine first. Right now, typical benchtop techniques, even when you get into the more sophisticated stuff like robotic pipetting, it lacks the ability to run drug discovery and compound discovery at the needed throughput. For example, in our current pandemic, what we're seeing is that people are getting infected with SARS-CoV-2. They're becoming healthy again, and people have started to talk about plasma treatment, harvesting their B cells, their lymphocytes that have this neutralizing antibody in it, and kind of just whole injecting it into newly infected people and patients. And so what I developed is a system. It's called Prescient, Platform for the Rapid Evaluation of Antibody Success using Integrated Microfluidics-Enabled Technology. It's a mouthful. That can screen for these neutralizing antibodies at an unprecedented speed. And now, why is that important for the Department of Defense? Is that they have many different applications in which they would like to screen different, whether it's cell candidates or for whatever applications they're looking for, and they need this screening to happen, on the scale of millions to tens of millions to hundreds of millions, which if you're, a typical benchtop scientist, it could take someone working around the clock 54 years, as opposed to you run it through one of these devices and it takes a couple days.
Okay, I want to stop you there. You said a lot of things that I did not understand, a lot of buzzwords that went over my head. So talk to me like I'm a 5-year-old.
Okay.
Let's start with, what is the coronavirus? What is a virus? And how does it work?
So a virus is, essentially, you wouldn't classify it as a living thing. It's not like a bacteria that, survives on its own. A virus needs a host, similar to a parasite. For coronavirus, what that is it's a special RNA virus. There's many classifications of viruses. Some common RNA viruses are, influenza. So it's probably, you would describe it as like the cousin to the common flu, maybe even closer, like sister or brother. Mm-hmm. And so, the word corona means Latin crown. You've probably seen a lot of these 3D representations of the virus. It's called crown virus, coronavirus, because the outer coating is filled with these kind of spiky horns, we can call them, that look like a crown. And those are the actual spike proteins that the virus uses to get into a host cell, which It could be a lung cell, could be a nasal epithelial cell, something of the sort.
And when was the first coronavirus created? What creates the virus?
Well, this virus is probably a host to host when it jumps zoonotically between animals. Viruses, especially RNA viruses, are susceptible to point mutations, where they kind of just recombinate. They change a little bit. And so this virus is probably just, a mutated past coronavirus that we've seen, and that mutated in such a way that it, made it very highly contagious, highly infective. So that's what most likely happened here. Again, I'm just speculating. I'm an engineer and not a virologist, but
so people who are at the scientists who are trying to discover a cure for this, what are they working on? What are the tough questions that they're facing right now?
Okay, yeah. So there's two ways to approach that, right? There's treating the infection and preventing the infection. I think everyone in science agrees that treating the infection is going to be the easiest kind of first step. Can we come up with drugs, especially if there's any, you've probably heard of hydrochloroquine, it's on the news. If there's any already approved drugs that can kind of help mitigate the symptoms. There's some other candidates out there, and that's probably what's going to happen well before a vaccine. I'm thinking maybe in the next 6 months, they'll have, approved drugs that are labeled and marketed for treating coronavirus, COVID-19. Then the next is, you would actually want to prevent this infection. Vaccine is what you're looking at there. There's many different types of vaccines, approaches to how to develop them. And I think the closest bet is going to be this neutralizing antibody therapy, is actually harvesting neutralizing antibody and generating your vaccine, your immunotherapeutic from that.
I understand that.
Yeah.
What is a vaccine?
Okay, so a vaccine is it's a drug that you would prime your body with, kind of just inject it, and sometimes it could be something as simple as you take the virus and you beat it up a little bit so it's weakened, and then you inject that into your system. And with the end goal of any vaccine is just to get your body primed for producing the right B cell, the right neutralizing cell that produces antibodies to combat this virus. And so, a lot of times it is just like a weakened viral strain that they put into your body. What the goal is, these viruses have antigens, which are just like little receptors on the surface that the body will read and then react to. They'll create these neutralizing antibodies. And every vaccine's goal is to prime your body in such a way, prepare your body so that it sees that antigen, that receptor, and then creates these B cells for that receptor. And so that way, if you do come in contact with the real virus, your body will have had that memory B cell in it, and it's able and capable of defending yourself.
Got it. What do you see as the timeline until we actually have a vaccine in the mass market?
I'd say 18 months.
And who's gonna make it?
Which company?
Which company? Which country?
I've seen a couple kind of leaders. There's some cool ones like Inovio has a DNA vaccine that's in the market. I know, and I forget the name. It slips me. There's an Israeli company that probably, is it Gilead?
Gilead. That's what I meant.
Yeah. They're probably, Gilead is probably I would say, if you're betting, that was who I'd put my money on. And there's some other smaller people, but yeah, Gilead's powerful. They're kind of drug discovery, compound discovery department, and they're top notch.
And so let's change topics. So that's not actually what you work on day to day. You're not working on the coronavirus. You're working on. So let's change to what you're actually working on.
Yeah.
Explain it to me in a 30-second elevator pitch.
Okay, it's a device, an actual credit card size device that this platform that you run these experiments on super rapidly with these nanobioreactors. And what I do is I put a single cell inside one of those nanobioreactors, and it does a whole bunch of other automated steps that I've engineered into this platform. And then your end result is neutralized cells that are not infected. And then what you do is you recover that entire antibody-rich population and begin your drug discovery from that. Is that better?
Okay, yeah, I have a much better idea of what you're
Okay.
doing. So why is that important?
Because current technology doesn't allow for, in realistic timelines, it's a bottleneck. It takes too much time to find these compounds, to find these targets. And what I'm doing is I make these, I think Dr. Fauci said there are a bunch of high throughput systems, and we've been in this field for about 6 years, high throughput screening systems that can run maybe 1 million experiments a day. So if you're trying to think of a normal researcher, normal scientist trying to use a benchtop pipette and traditional table, I mean, you can maybe do 10 to 20 of these assays or experiments a day, as opposed to, now that we have this automated microfluidic system that's running 1 million in a day, you can imagine your chances of discovering something just went up exponentially.
Wow. So give me an example of who would use your product.
So right now we are gearing towards COVID, SARS-CoV-2 studies. We just got our university approvals, and so what we do is we run the same experiment where we put in we take someone's blood, we purify it for the lymphocytes, for the antibody producing cells, and we run it through the platform, and we challenge it with virus, and we try to pick out the ones that are neutralizing. And that's going to be important, because once we have that neutralizing antibody, there's a lot of other questions that can be asked that will provide important information, but I think the main goal for a lot of people is getting this neutralizing antibody.
Got it. So let me explain it back to you. You take someone's blood, you purify it somehow, you put it on your credit card disc that you've designed, and then you throw viruses you throw antibodies at the blood, or you throw viruses at the blood, and then you see which antibodies purify or actually cure the virus, and then you can take out those antibodies and then turn those into drugs.
Yeah, that's exactly it. Because these antibody producing cells will produce antibodies in these bioreactors, these nano bioreactors, and we're talking on the size of 100 microns in diameter. Think of a little ball, this 100 microns, which is maybe a half the thickness of your hair. So these are really, really tiny bioreactors that we're generating.
Got it. And this is, you're not only going to do this for the coronavirus, but for the hundreds of other diseases that exist in the world?
Yeah, diseases. You could do it for, even if you wanted to find out biofuel production, and you have an algae cell, and you have a bunch of different algae cells, and you only want to get the top 10 producing bioethanol. You would run it through this device. You'd take all the the high producers, and then from there you would develop your library of algae cells that now are superpowered.
Wow. So who else is doing this?
So this field is called drop. It's a subset. So we have lab on a chip is the top, and as you can imagine, the name is you're trying to miniaturize any lab experiments, any lab techniques onto a chip, a device. And there's microfluidics, just tiny fluidic channels. And then in this is where the device I'm talking about is called droplet microfluidics. It's a two-phase emulsion. Just, it's the way you create these nano bioreactors. And so there's probably about, I'd say 5 to 10 strong groups out there. Groups being research labs. And then there's several commercial entities, but they're not, there's only one company that's currently in the same field, and they're in Germany.
Got it. So are you doing this with your PhD program or the Department of Defense?
Started with my PhD program, and then the Department of Defense kind of saw, oh wow, this is a very powerful system. Do you think it would work on X, Y, and Z, miniaturizations? And I was like, yeah. And they're like, okay. So that's what I do. Whenever I go there, one, I'm setting up the lab because they're traditional, more microbiology. And so they need more of the engineering components and actual physical equipment that we have down here. And so we're ordering that, and get that set up. But my main, work will be there's maybe 7 or 8 ongoing projects, and it's try to get all those onto an automated chip system.
Wow. Sounds fascinating, to say the least. Do you feel like you're at the forefront of like the newest trends in the Department of Defense's chemical department, like biotech group?
Yeah, I think a lot of people. There's a lot of really really crazy cool work being done in so
Mm-hmm.
many different diverse fields, and the good thing is like the facility is kind of, it's just more than just the biotech group. So you kind of can listen in and whenever they have open presentations, see all the cool stuff that's being done in robotics and
Mm-hmm.
all sorts of defense
Yeah.
projects.
What's it like to work for the Department of Defense?
It's pretty cool. It's a, I think it does fall into, a lot of people say like government work, some things that shouldn't take as long, like approvals and whatnot. They take longer. Chains of
Mm-hmm.
command has to go through a lot of people's hands and desks and signatures. The positives are, the funding is great. There's almost, that's almost never a barrier to when if you need supplies.
Mm-hmm.
And they really value the scientists there and engineers. I
Mm-hmm.
think that's more important. They do really care about developing that cutting edge technology.
Yeah. Is the biotech group a team of 10 or 10,000?
It's small. I think now it's 30-ish
Wow.
people.
Wow.
Yeah. So yeah, it's pretty
Congrats.
small.
Congrats for
Yeah,
joining that. It must
yeah.
be really challenging. So let's look ahead. What's the after the coronavirus? What's next? How are we going to better prepare for the next coronavirus? And even talking about military warfare when it comes to using chemical agents, how is your team thinking about this?
Yeah. So again, yeah, biowarfare is definitely an application for screening. That's been one question posed: is can we develop a platform so that we can take soil samples or air samples, screen them before we deploy soldiers into a certain area, so that we know what pathogens are out there.
Mm-hmm.
And so yeah, that's definitely an ongoing area of study.
Mm-hmm.
Um, in terms of, I think a lot of things will probably change in society because of the
Mm-hmm.
coronavirus. I think handshaking, special attention being paid to not touching your face as much,
Mm-hmm.
maybe regular decontamination of things like cell phones, which I'd really never done until this whole thing started. Now I at least twice a day find myself like wiping down my phone.
Wow.
It's pretty interesting. Just slight changes to life. As we know it.
Mm-hmm.
But
Mm-hmm.
for the better, and it's good. It's like we probably were just spreading tons of germs, and for the common person, it's not going to impact you that much. But for that immunocompromised person, you could potentially unknowingly be killing a lot of people or a
Yeah.
couple people a year, and so that's just a good thing to keep in mind.
Yeah. What made you interested in getting into this world? I remember you in high school were a star lacrosse player who was like a typical bro, and now here
Yeah.
you are as a scientist. So what's tell me how you've adapted and changed to put on your this scientific hat.
Yeah. So I think everyone matures a little bit as they go on, but specifically, what for me was in my last 2 years, and then that 6 months in between grad school and when I got my bachelor's, I was working at for the startup at a Rice, and I had a really really cool mentor, Glenn, and he introduced me to this whole world, and then beyond this, he was like, hey, you don't just stop here. Look at what else you might be interested in, and it was really him. He really propelled my desire to get into this field and work hard. And he told me straight up how it was. It's a lot of work. There's some reward. Every once in a while you'll get to a point where you're like, oh man, I'm being appreciated, and then it's a lot more of like, oh man, it's 4 a.m. and I'm still in lab. This really sucks. But that's just the way it is.
Yeah. There's this guy named Peter Thiel who I heard him say in his interview. He asked people, "What's a truth that you believe that the mass market doesn't? Something like an insight that you think is true that others think is untrue."
Dude,
what do you think?
Good. That's good. It's putting me on the spot here. Let me think about this for a little bit of for a minute.
Or
I wanna,
apply it to the coronavirus. What is something that you hear on the news that people are saying is true, but you know that that's not happening?
for coronavirus. Um, I mean, I don't know if a lot of people don't believe it. I'm maybe particularly more sensitive to it, that I see a lot of non-scientists trying to tout their scientific knowledge in ways that just, don't make any sense to me, and they're quite frankly very irresponsible. Specifically saying certain remedies, certain drugs are going to work when, there's just no backing for it. The science is always operating in a very meticulous, systematic manner. Some sure it can take a lot longer, but the good thing is it's preparing for certainty, right? We want to be 99% sure, 99.999% sure this is going to happen X amount of times. And I think a lot of people are kind of trying to undercut that in recent events to, take shortcuts. And as scientists, there's virtually, there's some ways to make your life easier, but taking shortcuts is never in the long run the way to go.
Okay. When I talk to people, we're talking about how in July everything's going to go back to normal and it's going to pick up. Is that ridiculous to believe, or do you think there is some credibility to that?
I think there's credibility to that. I've always been, I definitely agree with social distancing, physical distancing aspect. That's the most convenient way, or not convenient, but the most effective way right now. I do think once treatments are coming out, drugs for treating symptoms, people are wearing masks now. I do think by July we should, as a country and as a community, individual, because this will really be community by community basis. Everyone should be prepared to and ready to implement whatever strategies so that we can open up the economy again. People can get back to work and still be safe, right? Like, even if that means wearing a mask now for if you go out, even if it's just grocery shopping, just always wear a mask. There's going to be small sacrifices. I don't think July is too much of a stretch. I think, it'll be interesting to see what kind of travel restrictions are placed and whatnot. But I thought, I was like, okay, by the end of April, we should be open. We'll see if June, if it mildly opens, I think they will be doing it in phases, kind of small at the beginning and then kind of opening it up.
Is the purpose of a mask not only to touch your face, but to actually prevent the flow of
yeah.
Does it work like that?
Yeah, so it really does help you not touch your face. Well, not touch your face in the critical areas like your mouth, your nose. Your eyes are still pretty open, so keep that in mind. I think for everyone, it's just good practice to not be rubbing your eyes with dirty hands, regardless if there's coronavirus around. I think the masks are really good for, especially, surgical masks and just cloth masks, at keeping anything that comes out of your mouth or nose to yourself. It's not, I think unless you have the N95s or the, better than those, you're just not going to really prevent virus from coming in. And I do think coronavirus, your main danger here is going to be communication. It's going to be like these talking or coughing or sneezing spread. I really would like to see the more done just to tell us, like, how is it living on surfaces or aerosol? If it's really just staying in the air for hours at a time, that's really unsure. A lot of these studies, they stop. Their endpoint is, can they detect amounts of RNA? And so they're kind of just saying, is it there, as opposed to, is it there and can it infect you? Which is, I think, obviously the more clinically relevant question is that, is it there and can it infect you?
Interesting.
But it's just too quick. They need more time.
So who are 3 people that you recommend that we follow? Who are thought leaders in the space? Obviously, you got Fauci, but who are other people that people don't know about that you think are leaders, experts in the field?
And Fauci, Bill Gates. I need to think of one more. I know, I would say, in just technology and innovative thinking, a great person would be Steve Jobs. I always read him, but unfortunate. So let me think one more. There's
in regards specifically for learning about what's going on with the coronavirus.
Okay. I would say, I mean, everyone's okay. Well, a local person for me would be Dr. Julianne Leibovitz. He's a collaborator, and he's kind of one of the go-to people for coronaviruses in Texas, and he's a personal also project mentor for me, and he's just one of the most knowledgeable people I've ever met.
Yeah, wow. All right, so before I let you go, Joey, what are some last words?
No, nothing really of substance. Just as it's great to chat with you. It's great to see your face. It's been a long while. I see you're hanging out and staying safe in New York area, right? New York State. How's your surroundings?
I mean, I'm here in Long Island, so it's on the news. It's terrible, but in day to day, it's fine. You have your house, you got your backyard. I don't deal with anyone unless I go to the grocery store, and that's it. I feel like that's the same story for the millions of other Americans out there.
Yeah, I agree. I think it's good. We still have a little bit of freedom. Everyone's complaining about stay at home. This is so hard. I mean, my parents retired and they now live in Spain, and they've been on complete lockdown for 4 weeks now, going on to 5. Where you can leave the house to walk your dog. They don't have a dog. You can leave the house to take trash out once a week, and that's it.
Wow.
So they're going stir crazy. But again, everyone's staying healthy and they're doing what you have to do.
Awesome, Joey. All right, well, I really appreciate you on the podcast. This is awesome. You're the first science-related person I've had on the podcast, and I feel like people can get some value out of this from hearing what you had to say.
Yeah. Well, I had a great time.
Yeah. See you, Joey. Good luck out there, right?
Later, Benji. Yeah, we will.